Tim wants an N1C background.
Tim does. I can email it to Tim.
Okay.
Greetings, everyone. We'll start in just a couple minutes as people continue to filter in. Oh, let's see.
David, are you okay if I put your email and my email address into the chat if there's any follow-up questions?
More than happy to do that.
Yeah, and then I'll put the normal N1C email address also in the chat if there's anything N1C related. Is that okay with you, Nicole?
Fantastic.
Yeah, great, thanks.
Maybe also, oh yeah, Tim has been renamed.
Well, we like him as Stephanie as well.
We have a new Stephanie.
If we could only have an army of Stephanies.
That is very true.
David, Marlene, can I give it one more minute?
Yeah.
Yeah, go ahead.
Okay, let's go ahead and get started. Greetings, everyone. Good morning, afternoon, evening. My name is Nicole Nolan. I am the operations manager of the N1 Collaborative. It's our pleasure to host Marlen Lauffer and David Cheerie today for our workshop, which has been made possible by their incredible efforts alongside our patient identification working group. So just very brief background. Marlen is a clinical geneticist at the Dutch Center for RNA Therapeutics in Leiden, where she leads efforts to identify suitable candidates for individualized antisense oligonucleotide treatments. And David is a graduate student at University of Toronto, where he focuses on defining the clinical and molecular genetic principles of screening rare genetic diagnosis amenable to individualized therapies. So Marlen and David, we are so incredibly thankful for your efforts, including organizing this workshop. And without further ado, I'll turn it over to you.
Great, thank you so much. David, do you mind starting your shared screen?
Yes.
Thank you. Fantastic. Yes, so David and I are also welcoming you again. And I would like to say that this work has not been done by David and I, but really by the whole patient identification working group. And if you click next, please. Thank you. This is some of us. So we have been expanding in the past year. So this was initially the group of us, but now we are way more. And the patient identification working group is one of the working groups for the N1 Collaborative. And our idea is that we help identifying patients that are eligible for N1 treatments, at the moment specialized on antisense oligonucleotides, but also later on generally N1 cases. And there is a big group of mixed backgrounds. So we have geneticists, we have clinicians, genetic counselors, ethicists, and we also have a lot of early career researchers who've been putting a lot of fantastic effort towards this group in the past years. And also we are hoping to continuing in the future. So David, if you click further, please. Yes, thank you. The idea of what we want to introduce to you today is our variant guidelines, but it comes basically from the question of who do we want to treat, or who do we treat best, which are eligible candidates, which are the candidates to start with, and who do we want to prioritize. And the patient identification working group has been working on 2 or 3 different kind of pillars where we want to identify suitable patients. So if you click next, please. Thank you. So there's 3 subgroups we have been forming and 3 kind of pillars that we've been working on. So one, of course, is the variant subgroup. So if you look to a candidate and to a patient individual who is eligible, you have to look into 3 things. So first off, the genetic variant is a good candidate for an antisense oligonucleotide treatment. The other thing you have to look into is the disease subgroup, in where we have diseases or genes that are eligible for treatment. So they have to fulfill certain criteria, mainly at the moment kind of limited into what we can deliver, so which type of tissues are affected. And at the moment it is mainly the brain, but also now we can look into the liver. There's also possibilities to locally inject into the muscle, but many tissues at the moment are not reachable. So this is something we're still discussing, and many people are working on how can we deliver to other tissues. And the last thing we always have to look into is the patient. So is the patient eligible? What symptoms does the patient have? How has the patient been progressing? And can we really develop a treatment that is giving a clinical benefit in time? And while the second and the third pillar are very difficult to define strict criteria for, because there's so many things to assess, and these are often made on an individual basis, like really looking into a patient, how have they been developing over time, what treatments have been tried, what has been working, what has not been working. The genetics is actually something that is more targetable. So we really can define criteria of which genetic variants are good candidates for an antisense oligonucleotide treatment and which ones might not be as eligible. And this is what we've been working on in the past 1.5 years, and where we have been doing a lot of work with the group. And this is also what we want to present to you today. So we've developed consensus guidelines on how we select candidates for antisense oligonucleotide treatments. And the goal is really to enable researchers, clinicians, genetic counselor in the genetic rare disease field to make an initial decision of which candidates, which individuals to prioritize for these treatments. And once you have made this initial assessment on the variant, if this is a good candidate, then of course the other assessments on the disease, on the patient itself have to follow. But there's many, many million of these rare disease patients out there that have gotten a clinical diagnosis, and we wanted to enable and aid in this first step of helping to identify which variants, which patients are good candidates to assess further. So if you click the next slide, please. Yes, so this is what we want to introduce to you today. So these are the N1C variant guidelines. They are already available for the past few months on medRxiv. We will give you the link later again, so you can read up on them. They're currently in revision at one of the journals. And we want to introduce them to you today, and we want to guide you through the guidelines really practically. We give you some practical examples, and we also encourage you throughout the workshop to ask questions in the chat or raise your hands. So we'll give you a few examples, we will go through them, and we will give everybody the chance after each example to ask questions, because David and I really want you to be able to follow what we are doing and how we are working. So this is the aim of today. And David will now introduce the guidelines to you and lead you through the first few examples.
So this is going to be a brief introduction on the guidelines because they are a very detailed document, and we will have the chance to go into more detail with a few of the examples today. But I want to begin by saying that this was a multi-site effort. It was a collaborative effort. Multiple institutions and multiple experts in the field at all different stages within their career contributed to the development of these guidelines. So again, it's with their work that helped us develop these guidelines. So I just also want to point out that the guidelines were developed in multiple rounds of revision and piloting. So we drafted out the guidelines. We sent them out to volunteers, again, different institutes at different levels in their career, and they provided us with feedback, and then we applied that feedback, and we did that multiple times to get the final product. And in the final product, our guidelines allow for the assessment of 4 ASO strategies: splice correcting, which is what we saw with Millicin, where we correct aberrant splicing, which is the pathomechanism; canonical exon skipping, where we skip the exon either containing the pathogenic variant to get a truncated protein product that's still functional, or we could skip exons to allow for the variant to come back into frame, again, producing this truncated functional protein product. We could also use ASOs for transcript knockdown. This is useful in cases where the pathomechanism is gain of function or dominant negative. And then upregulation from the wild type allele. So if there is a wild type allele available that we can target, we can use ASOs to upregulate that in cases of haploinsufficiency. So again, our guidelines go into more detail about into these strategies and how we could classify these variants or these variants for eligibility towards ASOs on a scale. So the guidelines themselves are divided into 5 steps. Step 0 is a basic variant check. Is the variant you're provided correct? Then you go into the assessment of the inheritance pattern. How is this specific variant or this disorder inherited? Has the pathomechanism been described? These two are very important because we want this information to determine what kind of ASO strategies we're going to pursue. We also encourage our assessors to take a look at the effects of splicing. Does this variant cause a pathomechanism through splicing? If that is the case, this is a very ideal scenario to be using ASOs in, because we could use ASOs to correct that aberrant splicing that's causing the pathomechanism. And then lastly, we guide our assessors to a section in the guideline that directs them to other points of the guideline. And this allows us to pinpoint the sections within the guideline that are most relevant to our assessors. And I'm going to show you what that looks like. So in our guideline, we have this figure. Again, it shows the flow of the overall document, where we do the variant check, we check the inheritance pattern, the pathomechanism, and the effects on splicing. And then when you collect all this information, you go to this step 4 here to identify the relevant sections you want to direct yourself to. And depending on the pathomechanism and the inheritance pattern, we have different sections within the guideline that goes into greater detail on how you could assess that variant for that specific ASO strategy. So if we know our variant is a loss of function pathomechanism and it's inherited in an autosomal recessive manner, we go to section A and we read more on exon skipping. So this is what we're going to go through today with the examples. And once we assess the variant and we collect all the information we need, we are able to classify the variant for ASO eligibility on a scale. So clearly, eligibility towards antisense oligos is not black and white. They are going to fall on a spectrum. And this will help us identify the candidates that are stronger and those that we could probably pursue therapy development for. So on this spectrum, we have those that are unable to assess. This is not on the spectrum. These are just variants where our guideline does not apply to them. So this could be variants that are not diagnostic. It could be mitochondrial variants. This is if the inheritance pattern and the pathomechanism is unknown. Those are considered unable to assess. And then we have those that are not eligible. So these means there's either functional evidence that an ASO will not work, or the molecular principles or criteria don't allow for an ASO to work. And then on the opposite end, we have those that are eligible. So this means that there is functional evidence to prove that ASOs can work. And in the middle, we have this gray space where it's likely eligible and unlikely eligible. Those that are likely eligible means that the molecular criteria looks like a good candidate for an ASO approach. We just don't have the functional evidence to prove it. And unlikely is the opposite. We think that the molecular level, this is not going to be a good candidate for an ASO approach, but we just don't have the functional evidence to prove that. So again, all the variants are going to be classified on the scale towards splice correction, knockdown, or exon skipping. For those that are considered for a wild type upregulation, we cannot classify them on the scale just yet, but if a wild type upregulation approach does exist in the literature or in the clinical setting, we would consider it eligible. But for the most part, they cannot be classified on this scale. And again, we're going to go into a few examples. And in the guideline, what we use to help classify on this scale is these tables that we provide. So for each strategy, we provide a table. We provide the classification criteria, the classification and then the criteria. So this is specifically for knockdown ASOs. So you go through the guideline. Your variant is autosomal dominant. It's gain of function. You want to consider it for knockdown. And then this allows us, again, to classify it on that scale. So again, the most eligible are those that there is functional evidence to show knockdown is a good approach. And then those that are not eligible there's either evidence or there's molecular criteria that says that we cannot use a knockdown strategy for this specific variant or this specific gene. And then for those that are likely eligible, we know it's gain of function or dominant negative. And we don't think that haploinsufficiency is a cause of disease, or we know that haploinsufficiency is not a cause of disease. So we could consider for knockdown. But for those that are unlikely eligible, again, gain of function is a cause of disease or dominant negative is a cause of disease. But we know that haploinsufficiency is also a cause of disease. So we just want to be cautious about knocking down that specific gene. So again, this is all in the guidelines. It goes into a lot of detail on how to collect this information, what resources we could use. And today we are going to walk you through a few of these examples. But apart from the examples that we provide today, we do have a few resources that we shared in the preprint. The first being practice examples. And these examples were the ones used in actually trialing our guidelines. So they were selected from the literature or from ClinVar, and we sent them out to those volunteers who helped develop these guidelines, and we got them to trial it. So within the actual preprint, we have the list of these variants, and we have the answer key, which is the consensus using our guidelines. We also have instructional videos, and the instructional videos, again, walk you step by step through the guidelines, showing you the different web pages and the different resources and how to go about assessing variants for ASO eligibility. And then lastly, we have the eligibility calculator. And this is a tool developed by Marlen, and it essentially walks us through the guidelines again. This makes walking through the guidelines much easier. Again, it's a lengthy document, but this prompts you to answer the questions and helps you proceed through the document in an almost systematic manner. We encourage the guidelines to be used alongside this calculator, obviously, because it does reference the guidelines, but it allows you to input the answer and allows you to proceed to the next step. One thing I like about this tool is that you are not allowed to proceed to the next step unless you answer the question. If you're not able to answer the question, that means you don't have enough information to assess this variant for eligibility. So this calculator allows you to do that in a systematic manner. So we are going to be going through a few examples today. The first one is going to be a video example. This is one of the videos that's available on the N1C YouTube page. And then we're going to go through 4 live examples, with the last one being an example using the calculator. So I think this will be a good time. Is there any questions before we proceed to the examples?
No, so far no questions, but for the audience, if there's any questions that come now up during the examples, please post them in the chat or raise your hand. So after every examples, we will make a short pause and you can ask any questions you have. Thank you.
So we're going to go into the first example. And again, this is one of the video examples that we provide on the YouTube page. And typically, this is how the videos open up. It's going to show you the variant that we are going to be discussing in that specific video. And every video begins with the essential step of checking if the variant is correct. And for this step, we do suggest some resources within the guidelines. The one we use is Variant Validator. And the way this tool works is that you input your transcript of interest along with the variant, and it will just show you the output. And one thing I like to do is check if the protein lines up with the protein that we were given, and in this case, it does. If the variant were incorrect, the warning would pop up. Here, there's no warning, so we know that the variant we are given is correctly annotated. Following that, the next step is to check the inheritance pattern. So what we like to do is use OMIM for this. So if you go into OMIM and type in the gene name into the search bar, it will take you to that specific page for that gene, and you see that all the disorders associated with SCN2A, they're all inherited in an autosomal dominant manner. keep in mind, this could also be variant specific. So take a look into the literature. Usually you are given the information when you receive the variant, whether it's inherited in an autosomal dominant or heterosomal autosomal recessive manner. So you have that information. If not, take a dive into the literature, see how that variant has been inherited before. In this case, we know that this specific variant is inherited in an autosomal dominant manner. The next thing we want to do is take a look at the pathomechanism. We could also look at the haploinsufficiency in this case. For this specific example, I'll be doing that later. But this is the part where we would take a dive into the literature, see what's out there, what information is known about this specific variant. So for this, I typically like to start with a ClinVar search. Again, this is one of the tools used or recommended in our guidelines. It's just a database that uploads all the variants that have been reported, along with their pathogenicity classifications. So if we take a look at ClinVar, we see that a paper in 2018 by Barecki et al. Actually classified this specific variant as gain of function. And obviously we want to see what kind of strategies they use to classify this variant. So what we would do is click on that, click on the associated paper and see what that paper discusses. And this is one of their figures from that specific paper. And we could see here that this is our specific variant. And these are the associated phenotypes with that variant. They're positive for early infantile, mediate, or early infantile severe seizures. And then we see here that according to dynamic action potential clamp experiments, that this variant is specifically associated with gain of function mechanisms. So with all that being said, we know that our variant is correctly annotated. We know it's autosomal dominant, and we know it's gain of function. With that being said, we can move on to step 4. In this case, we're skipping over step 3 because there's no evidence for splicing effects. You could take a look in the literature. It doesn't cause splicing. The pathomechanism is the gain of function. So in this case, we could skip directly to step 4. And step 4 is the identification of relevant guidelines. So again, we go to this table. I just zoomed in on that table. And again, we have the pathomechanism on top, inheritance pattern over here on the left. In our case, it's gain of function autosomal dominant. So we could either consider for exon skipping, or we could consider for knockdown. So with exon skipping, there's 2 approaches you could use. You could either skip the exon to cause it to form a truncated functional protein product if it's in frame, with hopes of getting some residual function of that protein. Or you could use exon skipping to induce nonsense mediated decay. In our case, we can't really do that because this is in the last exon. So it might be likely that nonsense mediated decay won't occur if we were to induce frame shifts by skipping an exon. We're going to go into exon skipping strategies in more detail later on. So for now, I want to focus on knockdown. So in our guidelines, there's a section specifically dedicated to assessing a variant for eligibility towards knockdown ASOs. And one of the key components of this assessment is determining whether haploinsufficiency is a cause of disease. Because again, if loss of function is a known cause of disease, we just want to be cautious about knocking down that specific gene, because there could be other phenotypes associated with a loss of function. So one of the resources that we recommend in our guidelines is the ClinGen database. They have a dosage sensitivity table, which is a great resource. It shows it's a manual annotation of different genes and whether there is haploinsufficiency, or whether haploinsufficiency is a cause of disease for that specific gene. So in this case, I just look up SCN2A, and we see here that there is sufficient evidence for haploinsufficiency being a cause of disease. It's not necessarily an exclusion criteria according to our guidelines, a knockdown ASOs can still be considered if the disease is caused by haploinsufficiency. But we have to start considering the phenotypes. Are they more severe? Are they less severe? That's beyond the scope of our guidelines. But it doesn't necessarily mean we exclude this specific variant for development of a knockdown ASO. And I just have that highlighted here. I also want to note that the PLI score could also be used as an indication. It's really not as good as manually annotated scores, like the haploinsufficiency score on ClinGen, but the PLI score is also a resource that can be considered. Typically, if the PLI score is greater than 0.9, we know that the gene is intolerant to loss of function variants. So I also want to point out this figure that was in the paper by Barecki. And here we see that in the pink red boxes, these indicate phenotypes associated with gain of function mechanisms. And in the blue boxes, these indicate phenotypes associated with loss of function mechanisms. And the phenotypic group associated with developmental and epileptic encephalopathies are indicated in this gray box here. So we see that both gain of function and loss of function are associated with developmental and epileptic encephalopathies. So again, we want to be careful in considering knocking down SCN2A because we know that loss of function is also a cause of disease. So if we were to look at our table, again, we have eligible, likely eligible, unlikely eligible, not eligible. This would be classified as unlikely eligible because haploinsufficiency is a known cause of disease. But if we were to do a quick Google search, if we just input SCN2A ASO into Google, we would see that an ASO has actually been developed for SCN2A. This is Prax 222, so it's elsunersen. It's an ASO that's used to treat gain of function DEEs. So there is clinical evidence that this ASO is working. It's being used in clinical settings. So we could actually go back and classify this this variant as eligible towards ASOs because it is being used in a clinical setting. There is evidence of it working. So with that being said, that's just one of the examples. It's available as a video example alongside a lot more. So if you're interested, feel free to take a look at the N1C YouTube page. And before we move on, Marlen, do you want to give the acknowledgements?
Yes, great. Thank you so much. We're going to stop with the presentation here because David and I are very brave and we're going to go live in a minute. Hopefully none of the tools will crash while we are trying to do this. But I would like to thank everybody who was involved in this effort. So that's not just the patient identification working group, but every one of our volunteer assessors. So there's, I think by now, 39 people on this paper. We would also like to thank all the families that have been kind of donating their variants to us so we could practice. I think once we collected that, all of us together have by now assessed over 1,500 cases. So that gave us the knowledge and experience to develop these guidelines. We would also like to thank all of the other N1C members who have been giving us input and feedback. And also big thank you to Nicole and also Steph, who have been fantastic in helping us setting everything up, the YouTube channel, all the tools and helping us with this. There is now the link to the paper, but we can also drop it into the chat again. And with that, we stop sharing. And if there's any questions right now on the guidelines and on the first video, please do raise your hand or say something. Otherwise, David and I will move on to the live examples. Okay, so there's one question already, David. So for variants that are both gain of function and loss of function, can an ASO be helpful?
That's a good one. So I feel like it would be helpful if we consider exon skipping. That's a potential strategy that's applicable to both loss of function and gain of function strategies. If that gain of function and loss of function pathomechanism is a result of aberrant splicing, we could consider splice correcting. We might want to be cautious about knocking down that specific allele if both gain of function and loss of function are associated with that variant. Marlen, I don't know if you have any comments on that.
No, fully agree. So, I mean, that's what we will show in a minute as well. So when we can correct splicing, we always try to correct splicing because then we get the canonical transcripts back. And yes, exon skipping can be applied to gain of function and loss of function variants. We just have to ensure that the resulting protein still has enough function to maintain a healthy phenotype. So I think that that would be the answer to that question. So it's not a no, but you have to carefully look into this variant and see if there are options for this specific case. Right. Are there any other questions at this point?
Marlene and David, if I may offer just a gratuitous congratulations and an apology, I will have to step out actually to go give a talk right now, but I did want to come to the first half of the workshop. But
Thank
have
you
a
so much,
fantastic
Tim.
live presentation. Thank you so much. Bye.
There's another question. If we can share the recording. Yes, the recording will be online, so we will download everything after this, and Nicole will make sure that you can find it on the N1C website. So no worries about that. And we thought about also posting it to our YouTube channel, so you have the recording of the workshop alongside all the variant recordings as well. Okay, great. And something else, if there's a gain of function that gives worse symptoms than the loss, maybe try allele-specific knockdown. David, what do we think of that?
Yeah, so allele-specific knockdown is definitely a strategy we recommend. So in the guidelines themselves, we do offer considerations on when you would consider allele-specific knockdowns. Just keep in mind that once you start designing allele-specific, your opportunity for developing these ASOs just shrinks because now we're limited to polymorphisms or the variant itself, and that makes it trickier to design ASOs.
But I think there have been recent publications. So we know of a KIF1A patient that has been published where allele-specific or allele selectivity has been achieved, and these knockdowns are working. And the patient, at least this one that we know of, is doing well. So this is definitely an option to consider.
Yeah.
Yeah, so Annemieke was indeed thinking of the KIF1A patient. Wonderful. Okay, cool. I think then we move on, and I'll keep monitoring the chat and let you know if there's any further questions.
Yeah. So this is ridiculous because I cannot see my tabs on the top because that little zoom bar. So I'm gonna have to do this. I apologize in advance, but that zoom bar is not being helpful. So we're going to go into the first example, and I'm going to be cheating a little bit because I again walked through these already because I didn't want to. Let's see if I could go back. So the all the examples are here. The first one we are going to start with is this TIM DC1 variant. And by the looks of the specific variant, it's a deep intronic variant. So we're going to be walking through that live. So I'm going to just go back here, and I laid it out on this document so it's easy to follow along. But the first thing that we want to do is conduct a variant check. So we're going to go do this live. What I like to do is just copy and paste this specific variant into variant validator. So I'm just going to click try it out. I'm going to paste the variant in. I want to make sure that all my builds are going to be the same. So I'm going to be using the HE38. So I'm consistent throughout this presentation, and just click submit. So the only error that's showing up here is that there's a redundant gene symbol. This just means that the transcript is corresponding to the specific gene name. That's why it's redundant. Typically in these annotations, you wouldn't include the gene name. You just include the transcript ID. But everything else looks like it checks out. There's no other warnings. Everything looks correct. So now that we know that this variant is correct, again, the next thing we want to do is check the inheritance pattern. And I like to use OMIM for this. I have the link over there, but I also have it open. So what I'm just going to do is type in the gene. And if we see here, it's associated with one phenotype, and this phenotype is inherited in autosomal recessive manner. Again, keep in mind, there could be exceptions. Specific variants can be inherited in different manners. In this example, we know that this variant is inherited in an autosomal recessive manner. So we're good to go. We know the variant is correct. We know that it's inherited in an autosomal recessive manner. The next thing we want to do is check the pathomechanism and splicing. These are steps 2 and 3. I just combined them because just by the variant annotation, that should be a hint. This is a deep intronic variant. It's likely that the cause of disease, if it's a pathogenic variant, it's likely that the cause of disease is because of splicing. So I'm just going to combine pathomechanism and splicing. And what I like to do for this is use ClinVar. Let's see if I already have ClinVar open up. Nope. So I'm just going to open up that link. And it's showing up on another tab. Let me move it onto this screen. So now with ClinVar open, again, I'm just going to copy and paste. And it takes us right to our specific variant. Again, this is our correct variant. It seems to be well annotated. There's quite a few entries with quite a few resources here. So I like to take a read through these entries. And then I also like to take a look at the associated papers. And as you could see, I already clicked on a few of them, but the one thing I like to do is take a look at the most recent paper, see what's being said about this specific gene and variant. So if you just click the PubMed ID, it takes you to the specific paper. And coincidentally, this one is for developing an ASO for this specific variant. And if we take a look at the abstract, we see that the protein analysis showed a complete loss of TIMDC1 because of this variant. So we know loss of function is the pathomechanism, and that they actually designed 2 ASOs, splice switching ASOs, or what we call them in our guidelines as splice correcting ASOs, to restore the normal splicing of mRNA. So if we were to open up this paper, there is sufficient evidence. I'm just going to quickly scroll through it. I will point out a few things later, but if we quickly scroll through it, there is sufficient evidence to show that an ASO is working at both the mRNA level and at the protein level. We are able to restore function using an ASOs. So if that's the case, and we are satisfied with the evidence in this paper, we could go back and classify this variant as eligible towards ASOs. So what I'm going to do is just go to our table of contents. And we see that in the evaluation of splicing effects, if we go to that section, when we scroll to the end, we see that if an ASO has already been developed and shown to work with available functional evidence at the protein level, which it has in that specific paper, preclinical data is sufficient, we can classify that variant as eligible. But for the sake of this example, let's continue going to see what we would do in the case that an ASO has not been developed already. So we know our variant is correct. We know the inheritance pattern is autosomal recessive. We know the pathomechanism is loss of function because that paper that we pulled up said it's loss of function. Now we want to evaluate the effects on splicing. So we could dive into that paper in more detail to see what's happening at the molecular level to result in that pathomechanism. So I have a few points on that paper that I extracted just for the sake of time. But we see here that this is the splicing mechanism. It was obtained from RNA sequencing data from the paper, which is very important because there's specific strategies that we identify as in our paper as good functional evidence and strategies that we identify as not so good evidence towards splicing. RNA sequencing data we rely on it. So this is a good strategy. And we see here that the TIMDC1 variant acts as a splicing enhancer. So this is causing aberrant splicing, leading to the formation of this poison exon. And the poison exon is either 80 or 98 base pairs in length, and it has a stop codon within it. So what this is causing, again, loss of function because of that premature stop codon. So what we can do is pursue a splice correcting strategy because the pathomechanism is a result of this aberrant splicing, the formation of this poison exon. And I'm just gonna scroll here. Again, here's more functional evidence to show that this specific variant is causing a loss of function. So these are the two patients identified in the paper as having as being autosomal recessive or homozygous for this variant. And we see that there is no protein being produced. So we know it's loss of function. We know that splicing is the cause of disease. There's the sashimi plots in the paper as well. So we see that poison exon over here in the middle for these two patients, but we also see that the neighboring exon is still intact. It looks like the splice sites are still intact. We just have the formation of this poison exon. So with all this information, we have enough information to classify this variant using our table. So again, we're going back to our guidelines and we're just going to read through it. So let's pretend they did not design an ASO in that paper. All they did was look for the splicing impact. Let's look if this variant is likely eligible. There is functional studies, one of them being RNA sequencing, which is good. And this validates alternate splicing, which we have. We have the formation of a poison exon. And our variant is intronic. So we are going to focus on this section. The variant is deep intronic, so it's not within 100 base pairs or 50 base pairs of the acceptor site and the donor site, respectively. And according to the sashimi plots, it doesn't seem that the branch points or canonical splice sites are weakened. So we have enough information to classify this variant as likely eligible towards ASOs. These next 2 points we could ignore because those are only relevant for exonic variants causing aberrant splicing. But that concludes our analysis of this deep intronic variant. If you come across a deep intronic variant, those are the best candidates for ASO, in my opinion. They're also the hardest ones to find. I've looked through hundreds and hundreds of variants and only came across 2 deep intronics. But these are the most likely to be amenable to ASO therapies. So I really like them. They're great candidates, but they're not the only candidates. And we're going to walk through some other examples. So I will pause right there because there could be questions. Marlen, I'll hand it over to you to see if.
Thank you. So far, only people telling you that deep intronic variants are great,
Yeah.
which you all know. But are there any questions on this assessment or this variant in specific? I'll wait another minute, and if not, we can move on. So far, no one. So I think you can slowly move on.
Perfect. So the next example we are going to look at is this GNP tab variant. It's a deletion variant from what we can see here, and it looks like it causes a frame shift. So I'm going to pull up that document again. These variants that are exonic are going to be a little trickier than deep intronic. Again, deep intronic are great candidates. When it comes to these exonic variants, it starts to get a little bit trickier. So the first thing we're going to do is variant validator. Again, I'm just going to copy over the transcript gene and the cDNA annotation. I'm not going to copy over the PDOT because I like to use that as a check to see if it lines up. So I'm just going to submit. Again, same issue, redundant gene symbol. The transcript is correct. That's why we have that redundancy, but it all looks correct. The variant lines up with the protein annotation as well. So we're good to go. It also coincidentally shows the exon that it's in. So I'll keep that in mind for later. It's an exon 19. So the next step, again, is to check the inheritance pattern. So with that, we're going to go to OMIM, and I'm just going to input gene name. And there's two phenotypes associated with this specific gene. They're both inherited in an autosomal recessive manner. One is more severe than the other. The mucolipidosis 2 is more severe than mucolipidosis 3. That's not really relevant right now. But we both know that these phenotypes are both inherited in an autosomal recessive manner. Again, you'll be provided this information likely when you receive this data. But if it's not available, take a look at what's known in the literature. So the next step is to take a look at the pathomechanism. And again, I always like to start in ClinVar just to see if this variant has been reported before, and if there's any information that is known, it will be in ClinVar along with some relevant papers. So that's a good starting point. You could go directly into a Google search or a PubMed search. I just like to start in ClinVar. So I'm going to go back to the homepage, input this, and here we have the entry. So this is a well-classified variant. It's likely that the variants you receive are not going to be as well annotated as this one, but this one has been found in multiple patients, and they seem to be well described. So you could take a read through these, see what's being said about the specific variant. Again, I like to go to the literature and then just click on the most recent one. Again, it's a preference. You could take a look at the titles that interest you the most. I'm going to just click on the most recent one. And it's going to take us to this, I believe it was a review paper. And it goes into more detail about this specific disorder. Our variant of interest is mentioned in this paper. This is not open access, so I have it downloaded, but I also have some relevant parts that may be interesting to us. So if we scroll through this paper, we see that mucolipidosis is caused by GNP tab alterations, usually frame shift and nonsense variants. And these are associated with the loss of the Gallen-Lock phosphotransferase activity. So it's likely that this is working through loss of function mechanisms, because again, we have a frame shift variant that's probably leading to premature termination and nonsense mediated decay. So that's why we could possibly assume that this variant is loss of function. If we scroll further throughout the paper, we see that RNA analysis has been conducted on patient fibroblasts, and that it showed that the transcripts with a premature stop codon are eliminated by nonsense mediated decay. So we do have that loss of function mechanism for our specific variant. And our specific variant is discussed in this paper over here. I checked. It does undergo nonsense mediated decay, and loss of function is a known cause of disease. So now we know that the variant is correct. We know it's inherited in an autosomal recessive manner, and we know that it's loss of function. Now we want to evaluate the effects of splicing. I do want to point out that there's no evidence for splicing effects as being the pathomechanism for this specific variant. And also, if you take a look at our guidelines, I have it screenshotted on page 23 in the version that's available on the preprint. If the variant is nonsense and frame shift, leading to loss of function, and the pathomechanism is loss of function, we wouldn't really consider it for splice correcting, because even if we correct splicing, we still get a nonsense or frame shift variant, and that's still going to be pathogenic. So what we recommend is that we could consider exon skipping, which is section A in our guideline, or we can consider wild type upregulation in the case of haploinsufficiency. This is not relevant to us because we don't have our variant is autosomal recessively inherited. So what we can do now is that we know the variant is correct. We know the inheritance pattern. We know the pathomechanism, and we know we cannot correct splicing in this case. So now we're going to move on to step 4. Again, our variant is loss of function. It's autosomal recessive. Our only option is exon skipping. So that's what we're going to do. I'm going to scroll down into my cheat sheet. And with exon skipping, our guidelines, let me pull up our guidelines. Again, go into great detail on how to go through this exon skipping. So I'm going to go to section A, which is exon skipping. And again, it walks you step by step through different resources and strategies. And the way we approach assessment for exon skipping is we look for the obvious exclusion criteria first, and then we go on to the little more detailed nitpicky analysis. But typically the exclusion criteria is that we cannot skip exons that cause premature termination or affect the reading frame. So anything that's in the first or last coding exon, anything that's in an out of frame exon, or anything that creates a premature termination codon when skipped, we cannot use exon skipping. There are some exceptions, again, highlighted in our guidelines, but for the most part, we tend not to skip those exons. So that's what we are going to do with this example. We are going to look at if it's in the first or last coding exon. We are going to see if it's in an out of frame exon, and we're going to see if the exon creates a stop codon when skipped. So what I like to do is use this tool, Exon Viz. I'm just going to close a few tabs on this so I can make room. But I like to use Exon Viz to begin. And this allows us to visualize our specific, now I'm going all over the place. It allows us to visualize our specific variant and its location within the transcript. So I'm just going to copy and paste. Again, this link is also found in our guidelines, but I'm going to copy and paste the transcript. I'm going to click show non-coding because I want to see these UTRs as well. And I'm going to click submit. And we see that our variant is in the 19th exon. If we remember from our variant validator, it also said it was in the 19th exon. And we see that it's not in the first or last coding exon. The first coding exon is codon 1. The thin line here is the UTR, and then we start coding here. And then same with 21, last coding exon, thin line is the UTR, and then here is the coding region. This is important because the first and last coding exon are not necessarily going to be the first and last exon in the transcript. It's possible that the entire exon itself could be a UTR and the coding begins in exon 2. So pay attention to that. That's why I like using Exon Viz. It's a good visualizer for that. It also shows whether an exon is in frame or out of frame. And it looks like with exon 19, we are in frame. The phase is 2-2, meaning that coding one codon is taking place over 2 exons. So there's 2 base pairs and 1 base pair from exon 19, coding 1 codon, and then 2 at this end, and then 1 from exon 20, coding for 1 codon. So that's something to keep in mind, but for the most part, like 18 and 20, they fit into one another like a puzzle piece. So we know if we were to skip exon 19, we are going to remain in frame. Another tool that we could use to check this is the UCSC genome browser. So I'm just going to open that up and bring it over. I'm just going to quickly show this and then copy over again the transcript and then click search. So again, these are all tools mentioned in our guidelines. There's nothing new that we're introducing here. But what we have here is the UCSC genome browser. We see our variant has been reported in ClinVar before. There's a whole ton of information that's shown here. You could play around with the settings. The ones I have open right now are the UniPro visualizer to see what's being coded for at the protein level by that specific region. Base positions are open, haplotypes are open, ClinVar variants I like to see, so those are open, but you could play around with these settings. So the thing I like about this tool, and I think it's because of Marlen's work, that if you mouse over the specific transcript, which in our case, this is our transcript, the 24312.5. If you mouse over it, you could see that it's an exon 19 out of 21. The codon phase is 2,2, like I mentioned, and that it is in frame. So this information is also available here. So now we know that the exon is not in the first or last, it's not the first or last coding exon, and it's not an out of frame exon. So we could continue with analysis. I did mention that it is a phase of 2,2, meaning that there is a possibility a new amino acid is going to be coded. So another way to check that is using ensemble. So this is the part where people get thrown off, but this is something you have to pay attention to, because if these two exons come together, 18 and 20, if they come together and we form a stop codon, we're going to be getting loss of function anyways. So there's no point of skipping it. So we want to be careful and make sure that that's not the case. So what I'm going to do is go into ensemble and hope that it works today. Okay, the web page loaded. And I'm just going to input my gene. I'm going to click the human gene. And then this is our transcript of interest. So I'm just going to click that transcript. And I'm going to go into exons because this will provide us the sequence information. And if you scroll down, you see the sequence of every single exon. So what I'm interested in is the sequence of exon 18. The two last base pairs are going to join the first base pair of exon 20, right over here. So what we get is AGG. So I conveniently have a codon table here. AGG, we get arginine. It's not a stop codon. Coincidentally, if in the wild type, it codes for the same thing, AGG with exon 19, so arginine. So there's no new amino acid being introduced. There's no stop codon being formed. So for the outright exclusions, we're good to go. We can proceed with analysis. So the next step is to check exon size. So in our guidelines, we use a cutoff of 10%. If the exon codes for more than 10% of the coding sequence, be cautious about skipping it. It's not an exclusion, not an outright exclusion, but just be cautious about skipping it because it does code for a big chunk of our transcript. So what we are going to do is check the exon size. And again, I like to use ensemble for this. So here we have the length of our exon. It's shifted for whatever reason, but this is the length. And we could see 168 base pairs divisible by 3. It's in frame, but it's 168 base pairs. And if we go up, we see that the total sequence is 5,720 base pairs of this specific transcript. So simple math, we just divide 168 by 5,720 and we get 3%. So this exon codes for less than 3% of the coding transcript. So we know that there's no stop codon formed. It's not going to cause a frame shift if we skip, and it's less than 10%. Now we get into the tricky part. This part is probably going to take the longest. For the sake of this workshop, I'm going to give you the cheat sheet, but there's some details in our guidelines that it provides more detail on how to assess whether an exon codes for an important domain. And there's other strategies that we could consider. Please read that. It's very important to know how you would approach this assessment. And if it takes the longest, you're doing it right because it is the longest, the lengthiest part of this analysis. But what I like to do for checking an important domain, the first thing I like to do is use the UCSC genome browser because it has those UniPro annotations. So I'm just going back to, perfect, I didn't close it. I'm going back to our UniPro. I already input the gene, or sorry, the specific variant, and we have it here. And if we scroll down, these highlighted base pairs are the ones that are deleted. So if we scroll down, we could see all these UniProt annotations. It doesn't code for an extracellular domain. It doesn't code for a transmembrane domain. If it did, it would show a bar underneath indicating that that's a transmembrane domain or an extracellular domain. But we do see here that it does code for a domain. So I'm just going to mouse over to see what that domain is. If it shows. Perfect. Stealth protein, conserved region. Like, okay, great. What does that mean? We know it codes for a domain. Can we still skip it? Is it important? Does it play an important role? Is there any key enzymes or key amino acids involved in the catalytic site coded for by this exon? This is when you have to dive a little more into the literature. And the first thing I like to do is go right into PubMed and start reading up on this protein. In this case, I'm going to do that. And I'm just going to put in a very simple search term. GNP tab exon 19. Let's see what shows up. Oh, perfect. Development of an ASO for our specific exon. This was not planned at all, even though I clicked it before. But again, this requires access. It's not open access. I have it downloaded. You could probably access it through your institutions. But in this specific example, they attempt to develop an ASO for the skipping of exon 19, and they go into a little more detail about what exon 19 codes for. So I'm going back to my cheat sheet over here, and I pulled out a few aspects of this paper that I want us to pay attention to. So in this specific paper, it notes that stealth domains 1, 2, 3 are harboring a high number of variants that cause mucolipidosis. And with the fourth stealth domain, which is the domain that exon 19 codes for, there's only been one missense variant at the time of publication that is associated with mucolipidosis type 3, so the less severe. And then they also noted that these 4 important amino acids are important for the catalytic activity of this specific protein, but none of these amino acids are found in our specific exon. So it seems that this stealth domain 4 is less important for the function of our protein. Just keep that in mind. This is just based off of what's being seen in the literature. We definitely need to follow up with functional studies, but what it looks like is that this is not really that important of a domain compared to the other stealth domains, which harbor these important amino acids. So with that being said, we have enough information to classify this variant for exon skipping. So again, that's all discussed in our guidelines, and I'm going to head over to the table for the classification of variants towards exon skipping. So the first thing is this eligible? Evidence that exon skipping does not impair protein function, shown to be functional at the protein product level. So people would see this paper that we pulled up and be like, oh, they already developed an ASO, it's eligible. That's not the case, because we specify that it has to be at the protein level. There has to be evidence at the protein level. This paper only validates it at the RNA level. So this means that all it's shown is that we are able to skip the exon. It does not mean that it's still functional. All it shows is that we were able to skip the exon. So we cannot really classify this as eligible because we don't have evidence at the protein level. Now let's see if this variant is, or this exon is likely eligible for skipping. It is in frame. It does not result in more than 10% loss of the coding sequence. It does not create a stop codon when skipped, but this is where we can't classify it as likely eligible because it does code for a functional domain. It codes for the stealth domain. So let's move on. Let's see if it's unlikely eligible. Again, in frame, does not create a stop codon when skipped, and the exon does not result in loss of 10%, but it does code for a single functional domain. But if we see in the not eligible section, the domain is not really functionally proven to be important. So all the important amino acids are not coded for by that domain or that exon. Again, it's not the catalytic site, it's not a dimerization domain. It's considered less important out of the stealth domains. So we could actually classify this variant as unlikely eligible. And that is it for exon skipping. Again, I gave the cheat sheet. This analysis tends to be a little more time consuming, but I did it in advance. So it was a little more straightforward. But I will stop right there to see if there's any questions about exon skipping.
Thank you so much, David. It's very nice listening to you. You're just flowing so nicely. It's really good.
For
That
the
makes
past
me a little...
hour.
Yeah.
I apologize.
You can lay back now and then I'll take over. But do we have any questions on the things David has been discussing, exon skipping, but also looking into the splicing? So there was one question in the chat on multiple genes that are involved. So I already said this is something we can't take into account at the moment. So we're only looking into monogenic diseases or monogenic disorders at this point, but it's something we might be able to address in the future. Are there any other questions at this point?
I had a question. This is Deb Ritter from Baylor College of Medicine. Are you, so the sheet that we're looking at now, are we able to access this sheet? Is this part of the documentation or is this part of the, where can we walk through this?
Yeah, so this is not part of the documentation that we provide. This specific example is in the video examples.
Oh, great.
Marlen, if we do want to provide them, we can.
Yeah, we can share our cheat sheets, but all the things that are in there are of course in the actual guidelines, right? So you can find these things back, but I might have to clean up my cheat sheet a little bit, and then we can surely share it.
Great.
Yeah.
Thank you.
Great. Are there any other comments or questions at this point? Okay, David, then I'll take over. Is that okay?
Perfect. It's all yours.
Yes. Great. First, too many screens open. Let me share the one that is actually relevant. So that's this one. Okay. Right. Okay. Cool. So we'll continue with a few other cases. So I'll give you one example that we will do manually, and then I will actually walk you through one with the calculator, because I think the calculator does make things easier. And David and I actually, even though we have the calculator, do many things still manually because we're so used to doing it manually. I think we have some shortcuts at the moment. But if you're new to this, you might actually want to opt using the calculator. It talks you through everything, and you just have to read up on the important sections of the guidelines. But yeah, I'll also have a cheat sheet, so that is not as nicely done as David's at the moment, so I'll clean that up and we can share it. But I'm going to start with one variant, and this is in a gene called N1C 13. And this is a variant that, as you might see immediately, is at the canonical splice site. So +1 and 2, or -1 and -2 are all the canonical splice sites. And that's where I think all of us that are doing these evaluations, we get scared because we don't like canonical splice site variants. So we like deep intronic variants or deep exonic variants that are causing splicing defects, but we don't like canonical splice site variants. And that's because if you have a canonical splice site variant that is destroying the splice site, there's nothing we can do at this moment to correct the splicing of this canonical splice site variant. However, if it's a canonical splice site variant that is destroying the splice site, but it's causing a gain of function as an effect, that's something you can consider for knockdown. Or if you have a variant that is causing loss of function, but it's a haploinsufficiency disease, you can still consider upregulation from the wild type. So we'll use this example. And we will still consider some other options. So also here, I'll take a copy and I first start with the variant validator as David did. Just a note on the variant validator, it now needs you to log in if you have too many variants in a day. So this might happen. So you can just log in with your institutional address and then you will still get the results just fine. But also here, it tells you that the G name is redundant, but other than this, this is a fine variant and there's no problem with this variant. I will also look now, what is the disease that's associated with it, what is the inheritance pattern. So we also go into OMIM and type in the and do 13, type in the gene name. And this is an interesting disorder. So I'm ignoring the one that's kind of in brackets because that's actually not something that's yet confirmed, but we have this mitochondrial complex 1 deficiency. So these mitochondrial disorders, they often show as neurological diseases. So this is actually very interesting disease to study. And as you see, the inheritance pattern is autosomal recessive. So that already means that actually the haploinsufficiency might not apply here, so we can't look into upregulation from the wild type, and an autosomal recessive disease is most often associated with loss of function variants. However, you can still check, because this is something you might want to do. There are a few autosomal recessive diseases that are associated with gain of function variants, so you can also type in the variant into the UCC genome browser to see if that has already been described or if there's any information on that. Ooh, okay, it's loading. Okay, so I actually have also different settings up as David. So if someone really wants to know if there's already variants out there and have been described, what you can do, you can go onto the genome browser, you can go onto recommends track sets, and if you go onto clinical SNVs, what will happen, it will load all the different kind of databases. So we'll have ClinVar, LOVD, HGMD, Mastermind from Genomenon. All of these things will start being loaded. So you can see that here, I have them in here and you can look into this. So I can go into this variant now, and that also links me to ClinVar. And we open ClinVar. So this variant has been described 2 times, and 2 times as a likely pathogenic variant. So many variants at the canonical splice sites due to the ACMG-AMP guidelines are often classified as pathogenic or likely pathogenic. But as you see here, there's no information on this variant whatsoever. So we don't actually know if anybody has functionally studied this. So what we can also then do, or what I check, I do go into PubMed and type in the variant there as well and see if anything comes up. Okay, so nothing if I do it this way, so I can try a different search term, so just the gene and the variant. But then if you look into it, so there's actually, I mean, I've gone through this, so you won't find anything on the variant in here that easily and directly. And what I then also do, last resource, when I'm really getting desperate and I can't find anything on the variant before I give up, I literally just plug it into Google. Just do this, click Google. So, I mean, now my Google algorithm might be a little bit spoiled because I do this all the time, so it already knows what I'm looking for and it filters for different types of things. But if we go down here, there's nothing here now. So also I can't find anything here. If you do a little bit of a deeper dive, you will find a paper that actually looked into this variant and that did some functional analysis. So I'll pull that paper up, and it's actually from our colleagues at Sick Kids. So they did a lot of analysis on these canonical splice site variants where they did RNA-seq on blood samples, and they looked into them, and you can find this paper. And then we can also type that in here. I accidentally already highlighted this variant, and doesn't close. And what you will see in the last part, this is the splicing outcome from the whole blood. It says no frame shift, no NND presumed. That means the variant actually doesn't have any effect. And I wanted to highlight this variant because we see this a lot, that these canonical splice site variants are often classified as likely pathogenic and pathogenic based on the ACMG AMP guidelines. However, we know that quite a lot of them are actually not causative. And what happens in these cases with these patients, before you just dismiss them saying the canonical splice site variants are actually not eligible for correction with antisense oligonucleotides, please check if you have any functional evidence that this variant is actually causing a defect and what the effect is. And if there's no information at this point, please classify this as unable to assess and say you can't. This currently is not a diagnostic variant, and then go back and talk to the families or the clinicians if there might be another cause for the disease. I've had multiple of these discussions in the past year. Now they are very, very difficult to have because, of course, they have been given a diagnosis and they have been told that this is their variant. So they are looking for options to work with this variant. But I think we all have to be honest with these families and also the clinicians that wherever they got the information from, classified this variant as pathogenic or likely pathogenic, that maybe for this case, there might be another option. So we just wanted to give you one example, and these are not rare. I have them multiple times every year where I get a variant and I look up the variant or I check papers on functional data and I find out that that variant might not be causing the phenotype in the patient. So it's not actually a disease-causing variant. Yeah, so this as one example. David, is there anything in the chat? Otherwise, I'll just move on to the next one.
Sorry, this is
Yeah.
Deb. I was looking up some of the information on that variant. So the row column T you obtained, that is from a paper, that a reference
Yeah.
paper? Okay, got it. Okay.
Yes. So this is actually one that David and Greg at Sick Kids did. So they did this year, and the paper will be in the, if we hand out our cheat sheets, it will be in there. And there's a paper that you can download. The problem is it's difficult to find it because these variants are listed in the supplements, not only for this paper, but for many other papers. So often the search engines won't show them. On the other hand, the other thing is there is no other information on this variant, so you can't say that this is actually causing the disease if you don't functionally know what it's doing. So that's a bit of a tricky situation. David, I see there's another chat question coming in.
Yeah, it's a comment. This is an excellent variant that highlights the importance of clinical RNA sequencing to provide precise consequences of disease variants. And we agree.
Yeah. 100% agree. So this is what we have been saying all along. And these are things that we also highlight in our manuscript and our guidelines. We really need to check functionally what the variants are doing, which is why we have another splice site variant as a second example. So, voila, losing it here. Yeah, so we call this a non-diagnostic variant and we just classify them as unable to assess, but I always recommend go back to the patients, go back to the clinicians or the diagnostic center and ask them, hey, can you please have another look into this case? So another variant also at the canonical splice sites, again, canonical splice site variants are not our best friends because we can't just correct the splicing at that site, but we can still check if there's something else we can do for these cases. So again, here we go into variant validator and validate the variant. Yeah, so also this variant is correct. Here again, we have the redundancy in naming the gene. And also we can put that into OMIM to check for the inheritance pattern. Oh, and now I made a typo. Okay, so this one actually shows up as autosomal dominant. It's associated with two different diseases, both of them neurodevelopmental or neurological diseases. So they are interesting diseases to look into for targeted treatment with these antisense oligonucleotides. At this point, we now want to know a little bit more about this variant. So what is this now functionally doing? So again, I will check the UCC genome browser, but what I will do before I do the functional analysis, I will also check for haploinsufficiency because this is an autosomal dominant variant. So now I open this. So unfortunately on my screen, somehow this doesn't load properly, but I'll just type it in and then you have to believe the result. So this is ZD1A. You can see the gene here, and also here you have sufficient evidence that this is associated with haploinsufficiency. So if the splicing site doesn't work out, we still have the option to look into Tango approaches and see if we can do something with upregulation from the wild type. But first, we go and check really what do we know about this variant, and is this really disease causing? So we saw with the first example that this is an important step to do. So let this load quickly. Yes. So this variant has also been described. So you can see it here, and we can click on it as well and open ClinVar. And now you will see that this has been described multiple times, and this time we have comments. So I like comments because in the comments you can see what people were thinking and describing. So there was a lot of information. And there's also multiple papers linked with this variant. So this is always nice because then we can dive into the papers and read up on them. And here you can see in this comment, in 2 unrelated patients with a neurodevelopmental disorder, so this disease that is associated with them. I don't know how to pronounce that. So Kummeling et al. Identified a de novo variant. So this is this deletion on the canonical splice site in intron 15. And it resulted in splice site alteration. So that's great. And here also say they did RT-PCR analysis of patient cells. So this means someone did some functional analysis and really looked into this variant, and we can look up. So I would not just trust what people are writing on ClinVar, but I would really like to go into this paper. So we can go into the paper and look this up. So you can go through this. I'm not going to go through this in detail, but I'm just going to highlight the section that is relevant for us. So I have, again, this on my cheat sheet. And here it says, the resulting product shows that a transcript produced in individual 15, so that's the one with this variant, differs from the wild type. And then the sequencing of this product revealed that the variant causes an insertion of 81 base pairs between exon 15 and 16, and then it shows that intron 15 is not spliced. So that tells us that really that splice site on that site is destroyed, and that they have confirmed the splicing defect. And if we go into our guidelines, I'm just going to show this again. So we have step try assessment of the splicing, and we have our table. Voila. I think I have to click this better again. Yes, so this is our table on the classification for splice correction. And as I already told you, these are not eligible. So we go here onto the non-eligible cases. Canonical splice site and branch points are destroyed. So that is a no-go criteria. We won't be able to correct it with splice correction. Or the other one that goes along this, the variant is within 5 base pairs of the canonical splice site. So both of these are true for this variant. So there's nothing we can do in terms of splice correction for this variant. But we know this is a confirmed splice defect. We know it's causing a loss of function, and we know we have an autosomal dominant disorder, so we can still look into upregulation from the wild type. And there's multiple things you can look into upregulation from the wild type. So you can look, for example, into poison exons that you can skip to have upregulation from the wild type. You can look into natural antisense transcripts. So these are transcripts that are kind of blocking the transcription of the transcript you're looking into. So you can downregulate those ones, but you can also do a lot of UTR regulation. So there's many options. And there's multiple papers that have been talking about these different options. And what we did for our manuscript, and what is also available for all of you online, we combined them all in one big overview table, so you can look up if there's anything that anyone has described in any of these papers over the last few years for a specific gene. So I have this option open. And with that, I would really like to give a big shout out to Bushra and thank her for the fantastic work she has done. So Bushra is at Sick Kids with David. So she has pulled all of this together. And what she has done is she also pulled down the PLI scores and the ClinGen scores for everyone. We also added more antisense transcript that we found in the last few weeks. And then she has these 4 papers, Mittal et al., Lim et al., Felker et al., and Leo et al., we describe them in our guidelines that have in their supplement these information on these upregulation from the wild type, and we all just combine them into one big table. And what she has done and how this works, she has described here on the information page. So you can look that up, you can read up on this. But this is a fantastic resource and I really like to use it, and I use it every week. So now I'm gonna put this into our list. And here we have set D1A. And okay, so PLI is 1, confirmation again, ClinGen, haploinsufficiency score is 3. So again, this shows that it's haploinsufficient. And then I'm looking into these different options like antisense transcripts, poison exons, nothing has been described. So it's not looking good for us. And there's a bit on the UTR. So UTR regulation, you can, for example, modify regulatory elements in the UTRs, or you can look into upstream open reading frames that you can block, and that would then in theory upregulate the wild type. Okay, but just because it says the UTR is a great target, I would always recommend to still look into this. So I like to go into Ensemble and just look, how does the UTR look like? So we do that for set D1A and see what happens. Okay, again, we click on the transcript that we're interested in and we go into exons. And yeah, we see the UTR is 1 exon and a little bit of the second exon. And this is a nice example of what David was talking about earlier. The first exon is not necessarily the first coding exon. So here you can see that in exon 2, we have the ATG, so the start codon. And the first exon is actually the UTR. But it also shows me that the UTR is very small. So if I wanted to design an antisense oligonucleotide, it might maybe actually not be the best option to design this for this UTR. You can also look into the 5' UTR. That's a little bit bigger, but also not the biggest 5' UTR we have seen. So I'm a little bit skeptical if this might be a good option, and maybe might not be the best candidate to start with. Like if you're looking to prioritize your patients and your variants, that might be something you don't necessarily know. But for the sake of it, what I also still do, I check if I have an ASO, or I can talk antisense oligonucleotide, and then set D1A. You can do that in PubMed as well, or in Google. I still check. I still want to know, maybe someone has done it. Maybe already someone has looked into the UTR and knows and has shown that this works. So then you might still have an option for this patient. Yeah, surprise, surprise, there's a paper by us. So we did this actually this year for all the KMT2 associated neurodevelopmental disorders where set D1A belongs to. We had a look into what options are available and if we can do this. And what we actually did, we had an overview table of the different options. So here's all the genes, and here are the options also as well. Can we do that for tango approaches or upregulation from the wild type? And also here said D1A. And we came to the same conclusion that the only thing you could look into is the UTR, but there is at this moment no indication that this might be a good UTR to modify, and there has not been any functional studies done if, antisense oligonucleotides would be eligible to upregulate the wild type in this case. And so this is all what I'm leaving at. It's as maybe not the good end of a story, but to show you that while the canonical splice site variants are not good for splice corrections, you might still look into other things and help these patients that have these canonical splice site variants. And with that, David, are there any questions at this moment?
We have one.
Yeah.
Can you use ChatGPT to see if ASO treatment is eligible? Do you have any idea how reliable that is?
Yeah, this is a very interesting question because we have had these patients contacting us saying they asked ChatGPT and ChatGPT told them that their variant was eligible, and in those cases it was not. So I would be very, very cautious. I understand the patients. I understand that they are trying to figure out, but I would say AI at this point doesn't have the knowledge and understanding of the function of antisense oligonucleotides and the genetic background of which variants are suitable. So I would not rely on these tools at this moment. Cool. Okay. I'm starting to close a few tabs here because it's, okay, now we have that working. So now I'm going to show you our calculator because I think that's a very nice tool and my bar is moving all the time, which I really, really hate. Okay, so this is the N1C variant eligibility calculator that we made this year. And well, we did use ChatGPT to program the calculator, so that it was good for. So I think you can trust ChatGPT if you ask them to make a code for you. I wouldn't trust ChatGPT to analyze variants for you, but I think in this case, it was very, very useful. And what you can do, you can start your calculator. We will give you a little bit of information. You can also have a link to the training videos, to the guidelines, to GitHub, to our manuscript. So these are all things you can access also through the calculator. And what we have, you can submit your variant of interest, and then you have an answer log. So whatever you click throughout the calculator, it will be logged. So we take the next variant that we have prepared for you. Oh, we have only 1 minute left. That's bad. Okay. Then I'll just click you through it and I will just give you the answers for now. So you have that variant and you can submit it and then it opens this answer log for you and later on you can download it with a PDF. And you can start the assessment and it's asking, is the disease variant the causing variant? So hopefully that's a yes. And it will also give you all the information on all the types of disease, all the types of genetic variants that are currently excluded. And if you have one of those, then you can't go further. But in our case, none of them are applicable. Oh, sorry. None of them are applicable. So this is a variant that is applicable to these guidelines. When you look into copy number variations, you have duplications and deletions of genes. These are assessed separately with the calculator because different rules apply there. But in our case, we don't have this. We have a termination variant, so stop codon. And now it's asking me, what is the inheritance pattern? So I'm just telling you now, this one's autosomal recessive. And it's asking me, what is the pathomechanism of the variant? And it is a loss of function. And it also asks what is the variant type. So that's different than the actual guidelines. The guidelines don't go by variant type, but the calculator does because it spreads into different branches of the kind of decision tree that's behind it. And we have a nonsense variant. And this now immediately sends you to exon skipping. So you don't have to actually do anything anymore. It takes all the information you gave it and it immediately sends you into the direction that you wanted to go and gives you only that assessment and that section that you have to look into, which is very nice. So you're not getting lost. And if you did everything correctly until this point, it will either direct you to the Tango approaches, to the exon skipping, to the splicing, to the knockdown. So you don't have to do anything. But we have a biallelic variant that's a nonsense variant and it's loss of function. So the only thing we can do is skip the exon. And if you actually were looking into this exon, and I think at this point we should show you our cheat sheet later on, it actually creates a stop codon. So if you click, do any of the following criteria apply? It's a yes. And then it says, okay, you can't do anything. If you create a stop codon, you can't assess this variant. And you can click finish, and then it will lock everything for you. It's like, okay, you'll get back to the start tab. And here is your assessment. So you have all your questions and your answers, and you can download it as a PDF and then kind of save it and keep it. And it has the date and the timestamp on it. So you can go back in a year and reassess a variant if you have more information. So these things you can all do. And I think with that I stop because we are over time. So sorry about that. But if there's still an urgent question, I'm happy to answer it.
Hi, this is Deb. So if we put our variant information into the calculator, and if it is eligible, What
Mhm.
happens after that?
Yes. So, okay. So maybe first thing, you can put it in there and it will not save it on our site. So we don't know anything about your variants. They are safe. It's just for you to log it and you can download the PDF afterwards. And then we don't save anything. If you have a variant that is eligible, please reach out to us if you want to. We can help you kind of decide which way to go now and who could maybe help you with the case assessments, because we still have to look into the other things. We still have to look into the disease and we still have to look into the patient and the clinical phenotype. If you feel comfortable doing these assessments on your own, because also in our manuscript, we have given more resources to do these further assessments, you can do it on your own, but please always feel encouraged to reach out to us and we can give you further guidance on how to assess a case and then either link you to labs that are already working on a certain variant or a certain gene or disease, or if you feel like you want to start doing this in your own lab or on your own institute, we also have resources for you to learn how to develop these treatments for yourself.
Okay, great.
Great. Any other questions or comments? Sorry for keeping you longer than planned. Are we good? Good. I would say thank you, David, again. Thank you, Nicole. Thank you, everyone, for listening. And we hope you could learn a little bit today. And we are very happy to hear from you. Please reach out with any follow-up questions. We are happy to give more advice. And please use the tools and let us know how it's going. And if you have any input or ideas how to make it easier for you or more, user-friendly.
Thank you, Marlene. Thank you, David.
Thank you.
Thank
Bye.
you.